Thank you, everyone, for attending the René Salazar speaker series hosted by LatinX at the Broad. And thank you for the Broad Institute for allowing us to do this series. Just a brief introduction for the Latinx community. So Latinx at Broad has a mission of building community among Latinx Brodies and empowering members of Latinx communities. So we host events regularly. Today we have also a movie viewing. And the purpose of the speaker series is to highlight Latinx and diverse researchers within and outside the Broad Institute. We want to foster collaboration and allow visibility and role models for diverse individuals. And we also want to allow opportunities to network and promote research from our community. Please feel free to post your questions in the Q&A and they'll be answered live, or you can just put it in the chat and we can ask the question as we go. Now, I just want to give you a brief introduction of our speaker today. So our speaker is Dr. Filipa Rijo-Ferreira and is an assistant professor of infectious diseases and vaccinology at Berkeley Public Health with a joint appointment in the molecular and cell biology department, immunology and molecular medicine division. And for her, malaria is a major symptom and it's a very focal area of her research, such as sleeping sickness. And she combines molecular and parasitology approaches, mosquito development, and immunology and neuroscience to ask questions about the role of circadian rhythms in the evolution of pathogenesis of parasitic infections. So her focus will be on circadian rhythms and how they provide an advantage to organisms from bacteria to humans. And this will deepen our understanding of the daily biology of parasites, and it'll enable us to design effective interventions. So take it away, Dr. Rijo-Ferreira.
Thank you. Thank you so much, Ricardo. And it's an honor to be here and celebrate with you this wonderful community. I feel like also the past few weeks have been of uncertainty in science and just kind of uplifting each other, celebrating our scientific achievements and paying it forward for the next generation is very important for me. So I'm very grateful to have the opportunity to share with you our research on circadian rhythms in parasitic diseases. We are, as Ricardo said, based at Berkeley, and we are mostly focusing on malaria, but you will hear a little bit about sleeping sickness too. So, life on Earth is shaped by circadian rhythms, and the Earth rotation really creates these very predictable cycles of light and darkness that shaped how many different organisms evolved ways to anticipate these environmental cycles to regulate our sleep-wake cycle, our metabolism, our immune response. This is all controlled by our circadian clock. Clock disruption itself is quite detrimental, either through genetic disruption or through environmental inputs such as chronic jet lag. It leads to higher incidences of cancer and metabolic syndrome. Also, half of the genes in our body are under circadian control, and 75% of clinical trials that tested time of day treatment showed benefits at specific times, either, for example, for lower toxicity of drugs or higher effectiveness. However, a lot less is understood about how the circadian clock regulates infections. In malaria, it's really a striking example. And that's because malaria's main symptom is its periodic fevers. We knew these to be a characteristic of this infection before we knew it was caused by a parasite. Since the ancient Greece, you had descriptions of a disease that had fevers that would come and go at certain times of the day that now we know are really a consequence of a synchronous bursting of parasites out of our red blood cells. So this is a phenomena that you have, these unicellular Plasmodium parasites coordinated in their invasion, their replication, and then the burst out of the red blood cells that releases millions of new parasites and triggers our immune system to respond with a fever. And different Plasmodium species have this cycle last either 24, 48, or 72 hours. So all multiple of 24, which is quite an interesting number when we think about the circadian clock. And so for the longest time, we had assumed that parasitic diseases that had this rhythmic phenomena were really a consequence of the host rhythms, because most of these parasitic diseases do not have any free living stage. But what if parasites had circadian clocks? Well, this would matter because it would change how we think about their biology, but potentially would open possibilities for interventions. So how could we test that? We can actually leverage a lot the biology of the host. The mammalian system is very well understood, and amazing tools have already been developed. So if we focus, for example, on the clock of the mouse as a model organism, you can simplify it by having two key transcription factors, activators, clock and BMAL, that are binding to the promoter of multiple genes and activate their transcription. And amongst the genes that they transcribed are their own repressors. This is the families of cryptochromes and periods, whose proteins then translocate back into the nucleus and halt the transcription. The clock and BMAL, they activate transcription of multiple other sets of genes that act on different pathways, and that's how the core is so central to mammalian physiology. But this loop is really intrinsic, meaning that it still lasts 24 hours, even in constant conditions, and that is the definition of an intrinsic clock. So during my postdoc in the Takahashi lab, I was able to leverage these amazing tools that exist in the circadian field, such as, for example, mice that lack two of these repressors, the cryptochrome 1 and 2. And these animals now, once without environmental cues in constant darkness, they lose all their rhythmicity. So their sleep-wake cycles become disrupted, their gene expression, and so on. And so we were able to take advantage of these mouse model and infect them with Plasmodium parasites. We put them in constant darkness. We waited for 5 days after we infected them, and then we started collecting blood samples every 4 hours to assess by RNA sequencing the parasite transcriptome. And that was to really ask the question, what was going on the parasite rhythms? Were the parasite rhythms going to be as rhythmic as they usually are in wild-type hosts? Or now in these arrhythmic mutant mice, are the rhythms going to be disrupted? And so once we analyzed the RNA sequencing of the parasite, we realized that, as expected, in wild-type animals, even in constant darkness, the parasite biology was highly rhythmic. And you can see here in this heat map, 4,000 genes that are being rhythmically expressed across the day. So expressed peaking once a day. And these are 80% of the whole transcriptome of malaria parasites. But interestingly, even in these arrhythmic hosts, there were still about 3,000 genes that were showing these rhythmic expression patterns, really letting us to conclude that the parasite's rhythms are intrinsic to the parasite and not simply a consequence of the host clock. This reframes how we think about this infection. Malaria is transmitted by a mosquito. So this is a three-organism interaction, the host, the parasite, and the mosquito. And each of them have an intrinsic clock. And how we are now trying to understand a lot more in detail how they communicate with one another. But on that original discovery about their intrinsic rhythms, we also start testing if these, reduction from 80% of the transcriptome cycling to 60%, if that reduction was exactly as we would predict from a known circadian system. And so what we know from other model organisms of the circadian clock is that at the populational level, if we are, for example, considering a group of cells, all these cells are showing as an average a daily rhythm once they are exposed to these environmental cues happening in a predictive and rhythmic pattern. However, once in constant conditions, because it's an intrinsic property, the rhythm still persists. But as you can see, the amplitude of the oscillation is decreased. And that is because there is a slow desynchronization amongst the cells that are encompassing this population. And here you have that representation experimentally. So here, this is an amazing tool, one of the pioneer circadian tools that have luciferase. Let me play it again. That have luciferase fused to one of the repressors period proteins or under his promoter, and you can see once a day luciferase, so light being expressed in these cells, and this is a slice from a tissue from the hypothalamus, the suprachiasmatic nucleus. But if you now dissociate the neurons of these 20,000 neurons, you dissociate them and you put them in culture without any external stimulus, what you can notice if you were to follow one particular neuron is this beautiful rhythmic trace. But if you now look at the whole Petri dish, you can see that there is some unsynchronized Christmas light going on, right? And that you can now also represent multiple individual neurons. And if you were to trace a line for their average, you would see this flat black line. So that is what is expected, is that each individual neuron has a clock, but if you don't have any synchronizing cues, they will not be rhythmically in sync with one another. So we decided to take again malaria infection of these arrhythmic mutant mice and follow what happens later, right? We knew that until day 7, they were very rhythmic, but what happens later? Are they also going to be becoming out of sync? And so what you have in this plot is representing one of the stages morphologically, and that will be the ring stage, so the beautiful one at the top. Sorry, my cursor didn't seem to work. So the first one at the top of that little illustration. And if you look at 9 a.m., you would see almost 100% of the parasites all in this stage. And as day goes by, that continues in wild type animals. But in arrhythmic mutant animals, that eventually stops happening. And John Abel from MIT modeled these with us and compared it even to the desynchronization rate that would be expected from other circadian systems, and it seems that Plasmodium is in par or if even slower at desynchronizing based on his modeling. So that you would have a decline in the rhythms at the populational level, potentially due to a synchrony between each individual parasite. But what exactly, so that led us to conclude that the host still matters, right, for some synchronization. But what would be those potential cues of the host that were helping parasites be in sync with one another? And there have been 2 beautiful stories on proposing that the food and the timing of the host feeding were the main cues. And that is because when they were testing, again, the ring stage type of plotting, when they were monitoring when parasites were at that ring stage at nighttime feeding animals, or in the opposite, forced daytime feeding, so swapping the time that the food would be available to mice, you would see a shift of the parasite timing, proposing that the timing of food intake is defining when parasites rhythms is happening. So we decided to test how important was really the food timing for Plasmodium rhythms. And for that, we actually were able to take advantage of a different feeding regimen, which we described it as spread out feed. So we would have animals access to food through an automated feeder system that it was actually developed during my postdoc by Victoria Costa Rodriguez, who's now at the NIH. And Victoria's system allows is not only to monitor exactly when animals are eating, but program when they can have access to food. And so we can then take advantage and have the food be delivered at a regular interval throughout the day, abolishing feeding rhythms. And so how we actually read this out is through behavior assays, which we call actograms. So we can monitor here on the left side, you have the white bars represented the day light window, and the black bar at the top represents the dark. And you have the multiple days of recording. And in black, you have the animal's activity by monitoring how much they go to their running wheel. And you can see that mice are nocturnal. Most of the activity happens at nighttime. And with her automated feeders, we can now also monitor when the animal is actually taking the pellet for eating, which is represented in red, which you can see it also coincides during the nighttime. But then in the right panel, you have now the actogram of the spread out feeding regimen in which during the first few days, the animals are allowed to do what they want. So they still eat and are active at nighttime. But once we impose the feeding regimen, they still choose to be active during the nighttime, but they have to now to take access, and they are taking now the pellet every 1 hour and 42 minutes. So they eat the same, but the pattern is clearly disrupted, which you can see quantified on the most right graphs. So once we imposed these, and then we infected them with Plasmodium parasites, we went to see what would happen to parasite rhythms. But not much happened, right? If you see on the morphological side, parasite rhythms were still very obvious across morphological assessment, but also at the gene expression, there were still about the same number of genes cycling in both these conditions, and the overlap was really massive. So very little was changed upon this spread out feeding, meaning that the timing of food intake was not a driver for the rhythms and potentially is something else that is being modulated by the timing of food and not the actual feeding itself. So this study led us to conclude that indeed Plasmodium has an intrinsic clock, but the molecular mechanism and the host cues that really help parasites be in sync remains unknown. And Plasmodium is not the only parasite that has an intrinsic clock. Actually, sleeping sickness, which is caused by Trypanosoma brucei, is another example of a parasite that has an intrinsic clock. And this was actually work that I did early on during my PhD. And it is a much simpler system, so I thought it was worthwhile telling you a little bit about it, because this parasite is extracellular. So we can even culture it in the lab without any host cells. And so that made it much easier to assess if it has rhythms, because we could monitor the parasite transcriptome without any host cues. And what we found is that indeed, even without host cells, even without environmental cue changes in constant condition, there were about 10% of the parasite transcriptome that it was rhythmically expressed. And most of these were regulating the parasite metabolism, as you can see here in terms of pathways. And that translates into 2 rush hours of metabolic activity for these parasites when we measure, for example, ATP levels in cells. And we tried to understand a little bit about like why and what could be the consequences of these rhythmic biology. And because a lot of the anti-parasitic drugs act on metabolic pathways of parasites, we decided to do, amongst different challenges, a challenge for treatment with drugs. And so suramine is a drug commonly used in the clinic to treat these blood stage sleeping sickness infection. And so we decided to do a dose response curve, but initiate it at 4 hours apart across the day. And what you can see here is that There is, of course, killing of parasites as you increase the drug concentration, but there are shifts in these dose response curves, translating into changes in IC50s of about 2.5-fold, depending on when you're initiating, which you can see here represented in that little pills showing the increase, the fold change. So overall, that two different parasites have clocks and that regulate their biology, but why? Why would parasites have evolved? Intrinsic clocks is still a big question. And we can speculate multiple advantages for parasites. Perhaps it's really to anticipate the nutrient waves. They're going to need to have their replication. For their replication, they're going to need to have essential amino acids. We are ultimately their source for nutrients. We eat at a certain timing. That could be important as an evolutionary pressure. Could be about the immune response. The last few years have been quite remarkable in identifying many different immune regulatory pathways that are under circadian control in terms of cells coming out of the bone marrow at certain times of the day, migration, and also phagocytic activity of many different aspects of immune system. And so that could be an evolutionary pressure for parasites to be rhythmic. Could be matching the biology to the vector time of biting. The two examples of parasitic diseases that I mentioned involve a vector that transmits the disease. If the parasite transmissible forms were to be around at the time of biting, they would most likely be more efficient at propagating the disease. Many of these hypotheses that we have, actually different projects in the lab that are guided by them, but we are also able to think about that beyond the fundamental aspect of being quite interesting, that perhaps the existence of intrinsic clocks in parasites could be useful for interventions. And that is the example of the treatment time that I just mentioned to you regarding suramine as a proof of principle, especially considering that a lot of the antiparasitic drugs have a short half-life. But we can also think about the parasite clock as potential therapeutic target, since there is going to be very little overlap, if any, to our own clock. And so that could be another source of opportunity. And in our lab, we are very interested in understanding the interactions between host, parasite, and vector. But the first question we tackled in our lab once we started was really whether the parasite clock modulated malaria transmission. And this is the exciting, for me, the exciting work we have now finalized that I would like to share with you. So we've been thinking about malaria transmission, we cannot ignore the mosquito salivary glands. So malaria starts with the bite of these Anopheles mosquitoes, usually during nighttime. During that bite, both parasites and saliva is injected. And the saliva of mosquitoes has this amazing pharmacological cocktail of proteins that modulate our own biology. It modulates our coagulation patterns, platelet aggregation, there's immunomodulation, and even modulates the mosquito host-seeking behavior. Once in our skin, parasites quickly make their way to the liver, where they would replicate and progress to the blood stage, where they'll be able to be uptaken upon the next blood meal. So the salivary glands, by harboring parasites and also the proteins that are going to allow for this efficient blood meal, are really key drivers of malaria transmission. So we focused on these tissues. So what we've done was to had mosquitoes feed on infected animals, so they become infected themselves, wait 18 days until the parasites reach the salivary glands of mosquitoes, and then start collecting samples every 4 hours for RNA sequencing in mosquitoes that are housed in normal light and dark conditions and in constant darkness. Once we sequenced for RNA sequencing, we were able to assess a lot of the biological functions of the mosquito themselves. And what we found was that about half of the salivary gland gene expression was under circadian control. You can see here, these heat maps, these are about 5,000 genes that are cycling, expressing once a day in both light and dark and constant darkness conditions in these mosquitoes. Many of the pathways that were being regulated rhythmically is, for example, glycolysis, which 9 out of the 11 genes that encode enzymes in this pathway, which are here all color-coded, were being co-expressed, usually peaking around sunrise. And empowered or inspired by our reviewer too, we also went and looked for the protein abundance across time, and we saw that some of the proteins in the glycolysis pathway were also being timely regulated. Later on during the day, so more around sunset, we noticed that there was this co-expression of a lot of the blood meal-associated genes, which you can see here as their profile in orange, peaking before night onset. And you can see that each individual gene profile very beautifully rhythmic once a day. And I highlighted also 2 different genes that we know are involved in modulating malaria infection and showing these beautiful rhythms. So these are the genes encoding the proteins that I mentioned that are involved in anti-clotting, vasodilation, and anti-inflammation. But what about the parasites? Parasites within these salivary glands have for a long time been considered quiescent. And that is also because in part we couldn't really know if they were or they were fully quiescent or just non-replicative because the tools were lacking. And so Inês Bento from Maria Mota's lab took the lead to create a thymidine kinase parasite that basically now allows for detecting incorporation of EDU analogs to track DNA synthesis. So the parasites lack these, and so that was one of the reasons why cell cycle studies have been limited in Plasmodium parasites. But now we were able to create this line and confirmed that they do replicate early on, which is what you have represented here on the red panel. You have the circles highlighting the parasite, and you see a lot of little red dots in their nuclei early on in the mosquito. But then once we look in the salivary glands, parasites do not incorporate this EDU, meaning that they do not replicate within a 48-hour window in which we assess these parasites. So they are really non-replicative during the salivary gland stage. But what about their gene expression? So what we find is that even though they are not undergoing cell replication, they are very actively transcribing their genes. As you can see here represented many of the genes. So you can see the raw data in gray and the circadian algorithms fitted colors over in red and yellow. So about 20% of parasite transcriptome is under rhythms of gene expression, and that is many of those 20% of genes are encoding genes that are quite important for parasite motility and transmission. And that's, for example, the case of circumsporozoid gene, which is the key target of the 2 current malaria vaccines. And you can see their profile here. So we've been thinking about then, if there are going to be rhythms of transmission, we could think first about trying to assess how exactly the mosquitoes are feeding and choosing that in the lab as well as in the wild. The mosquitoes choose to preferentially bite at nighttime. And we also were able to show that those that actively fed, they also are able to ingest higher blood volume when we monitor hemoglobin levels at nighttime. Additionally, the load of parasites in the liver, so the first organ that they go and infect, is higher if mosquitoes are allowed to feed during that nighttime. But this is a complicated and complex phenotype, right? This could be because either mosquitoes are injecting more parasites at night, or whether the parasites are able to reach or invade the liver more efficiently at night, or whether the host is just more susceptible to infection at this time. And so to further dissociate these different possibilities, we went and studied in vitro and take parasites directly onto hepatocytes. And we show again that transmission is also more efficient at nighttime. And so to further disentangle the contribution of a parasite clock and a host clock, we started doing mismatched infections. So taking advantage of inverted cycles of light and dark, we can have either infections from morning or evening in either matched or mismatched conditions. And what we see is that infection is more efficient in a matched situation at nighttime. So ultimately suggesting that both the host and the parasite clocks impact infection efficiency, and that perhaps, and proposing that perhaps there has been this coevolution of clocks between these three organisms to modulate malaria transmission. So altogether, circadian rhythms of host, parasite, and vector matter. And together they modulate the malaria transmission and pathogenesis. And there are many outstanding questions that we are trying to address. We are highly interested in understanding how are parasites sensing time, what are the evolutionary pressures and host cues that drive their rhythmic biology. How does the mosquito clock impact infection? And of course, on the parasite side, what are the parasite clock genes that regulate their beautifully rhythmic biology? This is a very interesting aspect of biology because circadian rhythms have been described in organisms across the tree of life, cyanobacteria, fungus, plants, and animals. But the activators and the repressors are unique. So there's no homology. They have been evolved through conversion evolution. And so we do not know what would be those for these early eukaryotes protist parasites. But we hypothesize that it's still going to rely on a transcriptional translation feedback loop because that is actually conserved across eukaryotes. But this is still very much under exploration. So I would like to finish with that perspective that we do think that the circadian clock is central to pathogenesis and transmission of circadian diseases, of infectious diseases, and that disrupting the clock is likely going to be disrupting the fitness of these parasites and potentially can open new ways for interventions. And I would like to finish with a call to action. And so just there are, of course, I've been telling you all about parasites, but there are amazing work and research being done in the circadian subfield of infection, showing that there is a lot of time of day aspects of bacterial load, viral load, depending on the time at which infection is initiated. There's also a lot that we don't know. And so if you're interested, I think this is quite an open and exciting new field. But also, even if you're not particularly interested in timing, if you can just take something from this conversation, is that keep track of when you're doing experiments because time matters. And finally, I would just like to thank my junior lab, especially Brianna Parrington and Rushlin Pascual and Inez Bento, who led most of the work I share with you today, and our funders. Thank you.
That was very interesting. I think we'll go straight into questions. But before we go into questions in the chat, I'm just going to say that there's going to be a survey coming around, and the survey is to get your feedback on the talk. So I'll drop that in the chat. But in the meantime, we have a question from Clarissa Schaefer. Did you test different Plasmodium species? Could their circadian rhythms explain why they cause different lengths of fever cycles?
Yeah, we have so in mouse models, the most rhythmic Plasmodium is the one we chose, and they have these 24-hour rhythms. But we are actually in the lab trying to test a little bit of that, like trying to look at different now human Plasmodium species that have different lenses and trying to understand a little further why would that be the case. We do not have an answer quite yet, but we are highly interested in the same type of question. I think it's one of the big questions, why different Plasmodium species have it done differently. Could it be that they are just slower at the replication and they are keeping track of when still being exposed to the same cues as the others, but just takes longer to their replication? We don't know the answer yet.
Second question is from an anonymous attendee. Really cool findings on food entrainment. Since the parasites are more prevalent in tropical regions, do we know if temperature can also entrain their rhythms? One night parasite host infection in mice, considering that humans are diurnal. Is it safe to say it will be day in humans?
Mm-hmm. Yeah, these are two very interesting comments, and I do have that preferred same idea of temperature, and that is because of many different reasons. I do think temperature is in part responsible for the defect here, even on that food timing. But we have projects testing that. Also, there has in my sleeping sickness project that I mentioned briefly, I didn't point that out, but that was one cue that we gave to the parasites in culture, and that worked. So there is a precedent also for temperature to regulate rhythms in parasites, which I think it is quite possible that it happens also to Plasmodium. In terms of the day and night aspect, and mice being nocturnal and humans diurnal, that is, I think you could anticipate that. I think you don't need to think about so much. If we stop thinking about day and night and start thinking about activity and rest of the host, I think it matches very well.
Interesting. I had another question. If you were going to develop a therapeutic or an intervention for this, how would you implement this into therapy development, this information?
Well, I do think, for example, there's could be two ways we can think of. For example, let's give the example of the suramine treatment that I mentioned for sleeping sickness, right? So the treatment to sleeping sickness currently is actually quite toxic. So if we can think about a time that you don't need to use such a higher dose and you still achieve the same effect on the parasite, that could be useful, right? So just defining when to give a drug would have potential impacts, either because there's less toxicity to it or that you can achieve better elimination of parasites because whatever the target of that drug is highly expressed at a certain time during parasite across your day, right? So that could be the two very simple, actionable ways we can think about these research having potentials in the intervention easily. But ultimately, long-lasting type of future goal would be if you are able to show what are the molecular clocks of parasites And if these are really central to their biology, meaning that they allow them to anticipate aspects of our own biology instead of simply responding to our rhythmic biology, but anticipating it, like other organisms have evolved to anticipate the environmental predictive cycles of their environment, we could then disrupt those clocks, right? And not allow them to anticipate, which will be detrimental for their fitness. If that is all true, then you can start thinking about having small molecules that also act on these clocks and disrupt their ability to anticipate and be less fit to infect us, hopefully tilting the scale to the host to better control them.
So we have two more questions. Clarissa Schaefer again. That was a super interesting talk. One more question, if I may. Do you have hypotheses on what factors associated with food uptake influence the parasitic rhythm? And what about other physiological modulations such as inflammation rate or sleep deprivation? Did you
Yeah.
test different lengths of circadian cycles, shorter, light, and darkness cycles?
Mm-hmm. So on the first part, so we have why would food timing shift parasite timing, but it's spreading the feeding would not disrupt rhythms. What we have noticed is that if you do the spread feeding intake, a few, and this is again a lot of the work we've done it on Plasmodium, but we also have done it with Victoria Costa Rodriguez for metabolism assessment of liver, right? And what we know is that once you give this spread out feeding regimen, a few, for example, hormonal patterns like insulin become arrhythmic, but a lot, or spread out flat. But a lot of hormonal rhythms still become very rhythmic with the normal activity. So meaning that the same profile, even if you're spreading, it still remains the same. Some shift. So I think there's going to be a very complex integration of that timing of feeding that is happening in the mammalian body that is not necessarily just about nutrients, right? And so we do not know. We are having now an approach to look in vitro to try to now test each of these potential hormonal rhythms, nutrient rhythms, that it's a lot less convoluted and intricate as it is in vivo. But in terms of like the second part of your question, could you remind me, Ricardo? Oops, sorry.
Sorry, yeah, second part of the question. So did you test different lengths of circadian cycles, shorter light and
Yes,
darkness cycles?
yes, perfect. And yes, for that, we did test by using, so we tested in vivo, also in our original story about this, taking advantage of other circadian mutants that have a longer period. So instead of having their rhythms be 24 hours, they have their rhythms be 26 hours, which actually about 5 days in constant darkness, they are now in opposite phase to the usual time because they are like waking up. Think about it, that they would be waking up later and later, 2 hours a day. And so what we find is that the parasites fully are synchronized to these long period mutants. We have now been doing in the lab also with short period mutants, and we are wanting to do that also in vitro. So for sure, they are very much plastic in their ability to integrate, at least in a normal range. In terms of just, like you mentioned, in terms of light and dark cycles, a very recent manuscript from Sarah Reese's lab was testing these with environmental changes instead of genetic changes.
Cool. Okay, so we have another anonymous attendee. A very interesting talk. Were you surprised to find that only 20% of sporozoite genes seem to cycle? This seems to be a smaller proportion than in blood stages or in other organisms you described.
Well, actually, I was not surprised. I was excited because The reason is that in the blood stage, there's also the parasite replication that is happening, right? And there is a lot of the gene transcription profile that is happening because of a cell cycle and not necessarily about a circadian cycle. So I was happy to now see a lot less in being rhythmically regulated in the parasites in the salivary glands where they do not divide. 20%, it is definitely a lot less than 80%, but it's actually at par, for example, of the percent of genes regulated in the liver. So our most well-studied organ is the liver, has about 20% of its transcriptome being rhythmically regulated, and it imposes a major effect on our metabolism, right? So it really, 20% is still quite a significant portion. I probably would say that in our body, there's about 50% of genes being regulated, but it's not necessarily in the same cell. It is like across many different tissues that anywhere in our body that you would have about 50. So it's actually around the expected number.
Interesting. So, sorry. Hmm. So if we don't have any other questions, we can move forward. Thank you again for this talk, and I find it super interesting. Have you applied it to other tropical diseases? Like Zika virus was very popular a few years ago, when I was in the Caribbean. So I was wondering if you like looked at Zika virus or if you looked at any other mosquito-borne illnesses.
Uh, I did not, and I do agree with you that this is quite interesting. And I would imagine that other vector-borne diseases are probably going to be having this pretty much predicted, expected time of day transmission pattern that people have not studied. We are not looking into it, but I would imagine that, and also another interesting thing is that, for example, Zika is transmitted by Aedes species, meaning that is a diurnal type of mosquito. So I think that would be quite interesting to look at the salivary gland of these mosquitoes, whether blood meal associated genes are now with an inverted expression profile. I would predict so based on their function and what they are being regulated to do. And I do agree with you that this could be quite useful to think about other viral infections. There are some studies that had been done, I think, in vitro in terms of viral load, meaning that the ability of the virus to replicate inside the mammalian cell that has been shown to be rhythmic. I think that is in many cases expected, right? If you know our own replication machinery, a lot of the pathways that the viral will need to hijack to establish within our cells is dependent on host. And if the host biology is rhythmic, there is an expected link that was observed, but it's definitely not been studied at all in the more mosquito side of things.
Super interesting. I think that's it for now. If we don't have any other questions, we can log off. But thank you once again for your talk. And it
Thank
was super
you.
interesting and it was really nice to see. And thank you again for doing this talk in light of everything that's been happening. It's very encouraging to see.
Thank you. Thank you so much. I hope you guys continue these. I think it's quite inspiring for a lot of people.